Well, good morning, everybody. I am really glad to be talking to you today about proteogenomics. I want to say that this is the first time we've delivered a lecture on this subject as part of the training program for honors. And this is really exciting to me because proteogenomics is a really new area for proteomics that has been gaining a lot of fire. So you're quite likely to see the term in papers now. But I wanted to share with you a lot of the things that we need to be careful about when we do talk about proteogenomics, because in part it's a new field, it's a bit of a wild west, and some of the claims that you see in proteogenomics may require a little second guessing, a little skepticism, in fact. So I hope that today I can teach you about what the promise of proteogenomics is, and give you some examples of what people have accomplished with it so far. Of course, since this is part of mycobacteriology, we're going to be focusing on the tuberculosis side of things. So I hope you understand this particular emphasis area. But proteogenomics has certainly had a huge adoption in the area of cancer biology. So a lot of the studies I make reference to will be focused on that and on model systems like E. coli or yeast as well. Alright, so I'm going to start with a quick definition of what proteogenomics represents. From there, we're going to move on to talk about reasons why gene expression values are not the same as protein expression values. Why might you have a lot of protein when you have very little messenger RNA? Why might you have lots of messenger RNA and a relatively small amount of protein? So we're going to try to talk through that subject. From there, we're going to move into one of the most dominant approaches used within proteogenomics, which is the expression of non-synonymous SNVs, single nucleotide variations. So we'll have a strong emphasis on what we've done with that in a recent paper. And after this lecture, we will have a practical where you are looking at these problems from the data themselves to try to answer questions for yourself about what should have changed when a peptide sequence changes. From there, we're going to go on to the subject of regions of difference, which is certainly important when we're trying to compare different strains of MTB, for example. So regions of difference will be a really important topic for us to consider there. Okay. Now, I realize that one of the very first questions you might encounter is just what is this proteogenomics thing anyway? So I've tried to knock together a definition. I found it rather difficult to find a very concrete one that's been sanctified by the Human Proteomics Organization or by the National Cancer Institute, nothing quite like that. There are lots of papers using proteogenomics without really defining it. So I'm going to just start with this idea that it's a field of biotechnology. And it makes use of DNA and RNA measurements from sequencers in combination with data from tandem mass spectrometry. And the goal of this is to make the interpretation of sequencing data better and the interpretation of proteomics data better. So it's not just a one-way street, that we get better ideas of how to interpret our tandem mass spectra because we've got sequencing data. It's also a case where we learn how to annotate better in our RNA and DNA data by using proteomics. So this combination of technologies, this interface that exists between them, is proteogenomics. By the way, does anyone recognize why I chose those two molecules at the bottom right? Can you identify either? At the right, we have what? It's a helix. That should be a pretty good giveaway. Somebody want to say it? It's a double helix. That would be DNA. Excellent. Great. And the molecule on the left is an example of what? You can give me the obvious answer. It's a protein. Yes, of course it's a protein. This is an example of a structure called a porin. In this case, one that allows sucrose into cells. So this is a case where we're going to play proteomics data off of DNA data or RNA data. All right, great. So on we go. So there are several different kinds of tasks that we frequently find get called proteogenomics. In fact, you'll find that some researchers say since genomic data is obviously preeminent, the field should really be called genome proteomics. You should feel free to pay attention to that or ignore it as you like. Every omics is basically a term of convenience to help convey a set of common technologies to the next generation. So proteogenomics so far is kind of winning out on this one, and we'll see how it continues to develop. So let's start with some of the most basic applications that we've used proteogenomics for. we have sequenced a genome, and we want to know where are the protein coding genes within this structure. Right? That's a pretty straightforward thing. So can we detect novel ORFs? ORFs. Does the term ORF ring a bell with anyone? Are you concerned about it having your voice on camera? I understand that. That's okay. An open reading frame, right? So when we have a region of the genome that is due to be translated, we find a start codon followed by a whole bunch of other things, followed by a stop codon at some distance from it. So when you have an open reading frame that's present within your genomic sequence, you would like to know, is this or is this not expressed as a protein? So doing this via proteomics is very sensible. And it marks some of the earliest successes that were produced in this area. In fact, that paper by Jaffe et al. From 2004 was really the first to introduce the term proteogenomics. And everyone else has more or less followed suit since then. Okay, next up. Refining intron-exon boundaries. Now, in mycobacteriology, this is not a huge emphasis for obvious reasons. But in the case of human proteins, we frequently want to know, is there proteomic evidence that this pair of exons gets spliced together? Do we have the correct bounds of where this sequence shifts from exon to intron and back to exon again? Those are things that we can answer through transcriptomics or through proteogenomics, because you may have a peptide that gives convincing evidence that some transcript does exist that brings these two exons together. Next, measuring the expression of variant sequences. And let me try to pin this one down carefully. We know from genomic sequencing that variant sequences exist, but there are a variety of reasons why a sequence may be coded for in the genome and yet not appear as a mature protein. To use a simple example, something like cystic fibrosis happens when a mutation in one gene, CFTR, creates a protein product that misfolds and subsequently is never shipped off to the membrane to do its job. Instead, it's degraded. So even though you might have lots of transcription for this gene, the amount of measurable protein for the faulty chlorine pump is much, much smaller. So this is one of these cases where just because the variant sequence exists in the genome does not mean it's expressed at the same levels, that the protein is present at the same level as the other allele in organisms like us. Okay, which raises the question, how do we compare messenger RNA and protein quantities? Now, quantitation and proteomics are sometimes kind of uneasy partners, but we do have good methods for doing measurement of protein quantities, and we'd like to be able to relate those to messenger RNA quantities. So people have done this now in a variety of organisms, and some very interesting results have come out. So I've given 4 examples here, published examples of people using proteogenomic technologies, in some cases even before the term was coined. Because remember, the 2004 paper introduced the term. Okay, so I understand that we spend a lot of time in molecular biology classes talking about the central dogma, right? We have our replication, we have our transcription, we have our translation. That's all fine and good. But we don't think so much about what destroys these molecules, right? We transcribe to produce a messenger RNA, but we have a messenger RNA degradation process as well. We generate proteins through the process of translation, but we also digest up those proteins. So for this, I'm going to talk about how we get rid of messenger RNA and how we get rid of protein. So let's start with this paper from from the Bienen group. We have proteins and we have amino acids, and you can think of each of these as pools. So we have some pool of protein present in the cell, some reservoir of proteins that currently exist, and we have amino acids, free amino acids, that are both the fuel from which we're going to construct new proteins, and which represent the degradation products of other proteins. So you have some total amount of protein that's possible for a given number of amino acids in a cell. So Under steady state conditions, I'm sorry, I know the label is quite small, you have a degradation process that's digesting up existing proteins that is in balance with the process of translation, which is of course tied to what's being transcribed at a given moment. So if you have an equal amount of degradation and synthesis for a particular protein, you're going to have a steady state. The number of copies of this protein is going to remain constant. If, on the other hand, you have increased synthesis, so the increased translation of this protein, with no change in degradation, you're going to deplete the number of amino acids available while you increase the number of protein copies that are available. Is that making sense? Okay. But what about the other case where the process of synthesizing these proteins, of translation, is unchanged, but we decrease the amount of degradation of that protein? Here, these proteins are not returned to the pool of amino acids through degradation, but the number of amino acids is going to be diminished because we have an an unchanged translation, but an increase in the degradation. So you can see that a change by increasing synthesis or decreasing degradation has the same effect, essentially, that the number of proteins versus amino acids is going to grow. We also have the possibility of contraction. So it may be that degradation is happening more than it was while there's no change in synthesis, or you may have decreased synthesis with the same amount of degradation going on. In this case, you're going to end up with more amino acids that are free amino acids that are available to the cell. So there's always this process of balance between creating protein and degrading protein. Now the same is true for messenger RNA. We've already talked about transcription, plenty I'm sure, as a process of producing messenger RNAs from nucleotides. But it is possible, well, it's necessary, for the cell to return messenger RNAs to the pool of free nucleotides. Because once you've produced all the copies of protein you want for this protein coding gene, keeping around a bunch of bonus copies of messenger RNA is not so very helpful. So for this, we normally have some sort of 5' cap on the messenger RNA, and we have some sort of poly-A tail in this case. This tail is actually part of the signal of this messenger RNA. So once we use something like a deadenylase to chop off all of those A's at the 3' tail, this messenger RNA has been marked for destruction. And that after this decapping, after the removal of the tail, we have a removal of the cap, and then we have these enzymes called exonucleases. Do folks remember what an exonuclease is? Do you remember the term endonuclease? Like restriction endonuclease? An endonuclease can create chops in the middle of a sequence. Exonucleases can chop in from the ends. So in this case, the process of messenger RNA degradation balances the process of transcription. You know, as I was putting these slides together, I found myself thinking that we never really think about this other process. We have all these synthetic pipelines that we think about, and not so much how they go away again. So being able to destroy messenger RNAs when they're no longer needed, and the ability to destroy proteins when they're no longer needed, is quite essential for returning those building blocks to the pool. You know, when I was a kid, my family bought us building blocks a lot. So, you know, we had this pool of building blocks from which we could build our castles. But once the barrel of blocks was empty, no more building, right? So at the end of the day, you always had to destroy whatever you'd built in order to have building blocks to create something new. The cell is no different in its behaviors. Okay, now I have a couple quotes here that I want to read you from, again, some early papers from 2002 in both cases. This is before the term proteogenomics came about, but they're both quite relevant. So from Pratt et al. In 2002, they did an evaluation of a bunch of different proteins to ask about their protein turnover rate. The phrase protein turnover is one that we frequently use to describe whether we're in a state of pool contraction or pool increase, right? To reflect whether synthesis is overbalancing the process of degradation on a particular protein. So you might think, well gosh, all proteins live about the same length of time. This is not, however, true. they found that for the proteins they measured, the average rate of degradation was 2.2% per hour, although some proteins were turned over at imperceptible rates, and others had a degradation rate of almost 10% an hour. Now, do all of you have bank accounts? Good, good. So you probably had the conversation with your banker about compound interest, right? So the idea is that we're not going to give you a lot of money for your interest rate. If you have a great interest rate, maybe you're getting 5% on your bank account. So what that means is that if you put well, I'm going to talk dollars, I hope you don't mind. If you put $100 in your bank account, at the end of the year, after that money's been sitting around for a year, you should have $105 at a 5% interest rate. Now, at the end of year 2, do you just add another 5%, another $5, just like you did the first year? No, you don't, because of the power of compound interest. The money you gained in the first year is also gaining money during the second year. So instead of multiplying that first $100 times 5% to figure out how much gets added, you must also consider a 5% gain on the 5 extra dollars you had. Okay, so this is not different than how cells work. So I want you to think about this, we'll call this our typical degradation rate, right? Their average was 50, for these 50 proteins, was 2.2% an hour. So I want you to turn that around in your minds and say, what fraction of the copies of this protein remained after 1 hour? Okay, 2.2%. So was the amount that was destroyed each hour. So how much remained each hour? Okay, they're thinking about it.
Don't you take 2.2% start 50 and that would be the amount that was...
Oh, no, that's not the right way. That's not the right way. I'm asking a much simpler question. If there's no change, no degradation whatsoever, 100% of these proteins would be retained. If in 1 hour we destroy 2.2%, what fraction of all of the protein copies would remain? 97.8%. Very nice. All right, so she's just subtracted 2.2 from 100. Great answer. All right, now let's think of this as a compounding interest problem. If we keep 97.8% of the copies of the protein each hour, to figure out how much should remain after 2 hours, we need to multiply 97.8 by itself to take its second power. So we have a compound interest problem. If each hour we allow 97.8% to remain, that percentage is going to have a smaller diminishment each time, but it's starting from a smaller pool. So what I did was a little bit of math. I said to myself, I want to compute a half-life. Do you know the term half-life for biology? It applies just the same as it does in radionuclides. Basically, we would say, how long does it take before this set of proteins made at this very moment that only 50% of their copies will remain. So you can solve this by saying that 97.8% raised to what power, what number of hours, would give you the number 0.5 or 50%. And in this case, the answer is 31 hours. So if this 2.2% degradation rate is truly representative, we can say that the average protein lasts about 30 hours in a cell. But it's quite different when we look at something like this. If you're losing 10% of your copies every hour, you only need about 6 or 7 hours before half of the protein copies are gone. So this concept of half-life is actually quite shocking. For half of the proteins to be gone after 6 hours is a very different scenario than for half of the proteins to be gone after 31 hours. Would you agree? So proteins don't just last the same amount of time. In fact, some of the crystallins that exist in your eyes are going to stay there until the day you pass away. Creepy and strange, but old proteins can develop all kinds of weird aging problems and you end up with cataracts, etc. So take good care of your eyes. They have to last you a long time. All right, so that concept is one that I think is a really important one for us to think about. proteins may be destroyed quickly, or they may be destroyed relatively slowly. And in some cases, that turnover rate, the rate at which they're destroyed, is vanishingly small. Okay, now my friend Tim Griffin published a paper back in 2002 that would also qualify today as proteogenomics. So insights into the perturbative effects on genes involved in respiration, energy generation, and protein synthesis were obtained that would not have been apparent from measurements made at either the messenger RNA or protein levels alone. So this is an important thing to think about. If you are studying the regulation of particular genes, and you look at only the messenger RNA story, or only the protein story, you're missing half of the action. So you really need to have both of those appraised if you're going to understand all of the expression effects that you're seeing. If nothing else, that should explain why is proteogenomics important. We need to be studying both in order to get the full picture. Okay, now we talked a moment ago about how messenger RNA can have its tail removed and have its cap removed in order to mark that messenger RNA for destruction. We do have some people in the department who work with other kinds of modifications, this time not to messenger RNA but to protein, that can do the same, that can commit a particular copy of a protein for destruction. So when you hear about ubiquitin, I always want you to think, marking for death. When the cell says this protein has got to go, one of its most common ways to do that is to mark it with a ubiquitin ligase. So when you hear Soraya talking about the E3 ubiquitin ligase that the Parkin gene produces, you should think to yourself, a ubiquitin ligase is going to stick ubiquitin onto things and mark those proteins for destruction. So there's an increasingly broad pile of research that comes on about this. We have ubiquitin, here marked by these little gray discs, but we also have something that's marked here in black, and that's sumo. It's a fun wrestling name for this modification. But there's been some discussion that when we ubiquitinate a protein, it is to mark it for degradation. This is probably the most typical way that ubiquitin functions. But it is possible to mark those same sites with sumo instead. And when we mark them with sumo, we tend to think of it as a regulatory activity. We know also that in some cases, we can stack multiple ubiquitins onto a protein all on these long chains. And in some cases, instead of marking for degradation, it's marking these proteins to be regulated in a way. And it may be that we can even make chains of sumoylations as well. So when we have these complex combinations of sumo and ubiquitin modifications on a protein, they may be used for regulation rather than strictly used for the process of degrading that protein. So although we tend to picture ubiquitin as a way to mark a protein for death, in fact it can also play a more complex regulatory role. Okay, now I wanted to talk about 3 papers that are out there already in connection with mycobacterial proteogenomics. And the groups that are represented here are pretty important. So we start with Dinashri Kelkar et al., publishing in 2011. Dinashri was working with a researcher named Akhilesh Pandey, P-A-N-D-E-Y, who's a very famous guy in the field of proteomics. But Dinashri was focused on discovering whether or not the Mycobacterium tuberculosis protein sequences and the genome annotation that predicts them was complete or not. What if we have some problems doing gene annotation in the mycobacterial genome? It has some properties that make it a little hard to annotate. We know it's a GC-rich genome, for example, and that tends to make it a little harder to find where all the genes should be. Am I getting the GC richness right? Good. Okay, so we have these problems that being able to do a good genome annotation in this GC-rich environment is a little problematic. So they said the first thing we're going to do is throw out the normal assessment of what proteins should be observed in Mycobacterium tuberculosis. Instead, they did what's called a 6-frame translation. This is one of the handy tools that lots of bioinformaticists know about. A 6-frame translation starts from DNA, and it says, I'm not sure which strand is the coding strand. So I'm going to consider genes in a forward direction and genes in a backward direction from the same DNA. That all makes sense, right? So we have the coding and non-coding can be flipped either way. So that's already 2 possibilities we have to think about. The next is that we need to consider which reading frame are we in. Does the codon start at this nucleotide? Does it start 1 further? Or does it start 2 further than the first letter in the sequence? So we have 3 different reading frames in both the coding and non-coding strand, and we're not sure which one is which. So this is called a 6-frame translation. We produce every polypeptide that we could potentially from the translation of this DNA sequence in this case. All right, so doing that process identified for them 250 peptides that were not expected to be in the canonical genes, the canonical protein coding genes for M. tuberculosis. They concluded that there's still some work to be done on refining the protein list for Mycobacterium tuberculosis. These 250 peptides map to 41 novel genes that had not previously been called as genes in the proteome. So they made corrections to 79 different gene models, and in many cases, in 33 cases, they extended the start site a little earlier than where the open reading frame had been thought to start. You know, if you've got sequence errors in your assembly, you may miss where the start site should be. So they found quite a few changes like this. Next, I would point to Olga Schubert, who worked in yet another very famous mass spectrometrist laboratory, Rudy Aebersold. The proteomics community is paying attention to what's going on in Mycobacterium tuberculosis. So Olga, with the Aebersold group, did a similar kind of study, again with Mycobacterium tuberculosis, identified 29 novel proteins. You can see that the numbers are not huge here. They're not saying hundreds of new proteins. And she also focused on quantification. So not just saying, is this protein expressed or not, but trying to come up with some sort of quantitative assessment about how much signal was achieved from the peptides of that protein. So that's a really nice measure. One of the papers I cited in early examples of proteogenomics was from Gami et al. In 2002. They did quantification of a whole bunch of proteins in yeast, but they didn't go through mass spec. So here we see the same happening about a decade later in MTB, but this time using tandem mass spectrometry. Now the final paper I want to talk about here is not Mycobacterium tuberculosis. It's not even the tuberculosis complex. But I think we can agree M. smegmatis is an organism we care about a lot in Mycobacterium tuberculosis research. So here we see that we have a paper by, I always struggle with this name, I'm sorry, Tess Potgieter. Is that okay? She's giggling at me. She's giggling at me. That's all right. I'm going to deal with that one. So Tess is finishing up his PhD over at the University of Cape Town. Yeah, he's right across the river from us, and he is doing some great work in the space of proteogenomics. And this paper from 2016 did the similar kind of 6-frame translation process for M. smegmatis, where he found 63 novel open reading frames. So it may be the case that although we think of the canonical proteins for each of these species as very well established, in fact there's a little more information to be found when we look at this through protein expression. Okay, now I want to talk about a paper from Tian Heunis and Anzan Dippenaar. Now Tian is no longer with us, he's moved up to Newcastle, but he's doing some really cool work, continuing to do great work in proteomics. When he was here, he worked with Anzan on 3 different strains of Mycobacterium tuberculosis. I think everyone remembers H37RV. You've heard that one endlessly by this point. But we also had 2 strains that were isolated in the local community. I'm going to call this one Saucy 3517 and Saucy 3651. These cultures were grown up multiple times, and from each of these we produced 2 technical replicate gel CMS experiments, which is a way to spend more instrument time to get a more comprehensive proteome from these cells. So we wanted to know, can we learn more about these cell lines by combining proteomics information and genomics information? Now this brings Anzan to the fore. Anzan was really interested in finding ways to play the data we produced off of a HiSeq 2000. These are sequencing reads in whole genome shotgun produced not just from H37RV, but from the clinical isolates from the local community, alongside these data that we had produced on the mass spectrometer. How can we make these two work together? Now, we started with a mapping exercise. I think you remember from the bioinformatics lectures that when you have a good reference genome to work from, an annotation, that it's pretty easy to just map the reads produced in whole genome shotgun against the FASTA reference that we have. So this produces an alignment. In this case, they were using 3 different aligners and trying to ensure that if one of them was giving anomalous results, it wouldn't taint the overall result. So many of the genome sequencing reads mapped correctly, and that could be used to determine if we had any sequence variants in the known genes of the creature. However, she also looked at the sequencing reads that did not align to the reference. And for that, she did a de novo assembly to try to determine which, do we find that there are bits of additional genetic material in these circulating isolates that are not part of the reference. And yes, indeed, she found that there were some pretty big regions that had not really been observed as part of the H37RV genome. So we were able to use 2 different kinds of references here. In one case, we have the canonical protein sequences from H37RV, and we add to the database sequence variants that are known within these protein sequences. So the same sequence database that contains the wild type sequence also contains the variant sequences that were detected through the genetic variant screening. But we're also able to add the unmapped sequences through 6' translation to our database to be able to detect those. So we're using a canonical database, a canonical plus sequence variant database, and a canonical plus variant database, plus the unmapped region database, all being passed to figure out which peptides could be identified from proteomics. So we would start with the fact that when we do this kind of expansion, when we add new sequences to our database that we have to consider, our search space gets bigger. We have more proteins in the database against which we're going to compare all of these tandem mass spectra. So here we've used a split axis, and I want to try to explain why. So here's the 4,000 odd proteins of H37RV. If we were to add sequence variants that were specific to SAUCY 3517, we see that we get just a few more proteins we have to consider. A few. It's not a huge number. Likewise, if we were to look at the sequence variants we need to consider in order to identify known variants that were detected through genomics in 3651, we have a few more than we did in the original H37RV. Once we add stuff like the unmapped regions, the 6' translation of the stuff that didn't map to the annotation, we have a pretty big bump in how many different sequences we must consider. But look how much worse it is to start from the H37RV genome and just 6' translate the whole bloody thing. The number of sequences we have to consider in this case is right off the scale with everything else. So although 6' translation is very frequently used as a basic tool for proteogenomics, we find that the impact on how many different sequences one must consider under those cases is pretty troublesome. So that's not an ideal way to go if we can avoid it. All right. Now, I know that everyone loves trying to interpret tandem mass spectra to sequences. We will get a good chance at doing this in a practical in a few minutes. But for now, I want you to think about the fact that we have different sequences for these peptides that are known to be present because of our work with the genomic sequencing. Remember, we sequenced these strings, thus we know particular variant sequences that should be found in this creature. So we can say that the sequence here should be, say, NGDEVQ, but here the sequence is NGDEIQ. You see that? So we have a valine or an isoleucine at this position. Either of those may be detectable depending on whether or not this organism has that sequence variant in it. But what we're able to show is that when we look at SAUCY 3651, we see an isoleucine. That's the sequence over here. When we look at H37RV, we see a valine at that location instead, the wild type sequence. If we had not included that variant sequence in the database, we would just have failed to identify the spectrum. So it's valuable to us to be able to incorporate genetic variation when we look in the protein sequences that we compare to our spectrum. Okay. Now, you might think that I'm telling you that the only thing you need to worry about would be variants of normal sequences that exist in the database. But in fact, we found some really interesting stuff when we looked at the sequencing reads that did not map to the annotation. You remember that we assembled this and then did a 6-frame translation from it. Did we find anything? The answer is yes. When we looked at the added region of genome that had been added in these sequence variants, we were able to find, in one case, a novel helicase. So when we have a helicase, we have a protein that's interacting with DNA. We see that there were 29 peptides. It's a huge number. I mean, this is not a phantom hit. 29 peptides were all seen from this helicase when we look at SAWC 3651. So although H37RV doesn't have this helicase in it, this variant that's circulating in the population has a whole new gene that has lots of peptides and evidence present within it. So what special abilities does that helicase confer on this clinical isolate? Does it make it better able to survive in the wild instead of in the lab? Is this a functionality that's been lost from the lab strain? There are lots of possible interpretations from it. What we can say is that this is genetic material that is missing from the H37RV that we treat as our canonical sequence, and it has additional genes that we didn't know existed. But now, through proteogenomics, we're able to identify it conclusively. Okay, just a moment. One of the terms that I found a little confusing when I first joined this department was RD. RD. People weren't talking about roads. They were talking, in this case, about a region of difference. And when we look at many different strain variants of Mycobacterium tuberculosis, whether they're Beijing or Latin American Mediterranean or whatever, T-type, I mean, there are all kinds of different families of these Mycobacterium tuberculosis, would be that we try to determine which deletions are present in a given genome. And we use that as part of our strategy for determining what evolutionary tree of the circulating Mycobacterium tuberculosis that this represents. So I thought it'd be really valuable to go back to this 2004 paper. This is one of the early efforts, I'd say, to try to systematize all the different regions of difference that were known to separate different members of Mycobacterium tuberculosis. Now, I would note that when we looked at the regions of difference in the Heunis et al. Paper with Exon, some of the differences that we discovered, some of the deletions we discovered, were not part of this canonical list of RDs that we work with. So whenever you sequence a new strain, you're likely to encounter some deletions that are specific to just that variant. But in this case, we're looking across 68 different regions that sometimes get deleted in the circulating strains of MTB. You can see that some of them are ridiculously variable. The region here in that vertical stripe represents presence or absence in a whole bunch of different strains. I think they were looking at 100 different, I didn't include, okay, about 100 clinical isolates of MTB. So here they're asking, was this region deleted or was it not in this? You could think of this as kind of an alternative to spoligotyping, I guess, for classifying them. All right, so in this case, they asked how much of the total genome gets deleted in some bug or another. And what they found was that only about 4.2% of the total genome gets removed in this way. Now, you might imagine that if you were to nuke away proteins that play an essential role in glycolysis, that's going to be pretty lethal, right? I mean, bugs have to be able to do central metabolism, right? So what they found is that a lot of the regions of difference do contain genes in this case 224 of them for this set of 68. And yet, most of the genes that it removed can retard growth, but not kill the bug outright. So we can think of the functionality that these 224 genes represent as useful to the cell. They come at a survival advantage to have them, generally, but in some cases, they've been deleted nonetheless, and that they can continue to live. The other thing is that you shouldn't think that regions of difference are just scattered willy-nilly throughout the genome. They actually tend to be clustered together, that some regions are home to a larger number of regions of difference than other parts of the genome that are presumably much more critical to function. So I've used the term syntenic aggregation. Remember that we use the term synteny to refer to the physical ordering of genes on a strand of DNA. So we have aggregation among these regions of difference. They're not just spread uniformly throughout the genome. Yes, question?
So the 224 genes are not essential?
Right, yes. The bug is able to grow and to be pathogenic, in fact, even without some number of these 224. Now, if you strip out all 224, if you rip out every region of difference from a particular strain, it probably is lethal. But I would imagine that you can have either this deletion or that deletion, but not both in some cases. But generally speaking, yes, the genes that get deleted out of particular strains tend to be nice to have, but not necessary. I mean, would I die without my Starbucks coffee? Clearly not. I live in South Africa, right? But it's nice to have a Starbucks on occasion. Yeah. In the same way, the cells would like to have the functionality of these 224 genes, but they are not individually necessary. Good question. Okay. So here's a problem for you in proteomics. Proteomics is pretty good, it's getting better all the time, in fact, at saying something is present if that protein is really there. Right? You're going to get some sort of evidence back if that protein is present at a suitable concentration. Okay, so we can prove that things are truly there by proteomics. But can you prove a negative? If a region of difference exists in that strain, are you able to prove that that region of difference is in fact not expressed here? So I want to give you some cautionary tales about this. When we did this paper with Tian and friends, we asked, were any peptides detected that matched to regions, in this case a SOC 3517 specific region of difference, and RD152, one of the more canonical regions of difference. We asked, were there any peptides that could be found in those regions? And there were some. So how could a peptide be observed if the DNA that codes for it is missing? Right? That doesn't make a lot of sense. So we asked, why would we see those? And we answered that the hits we observed are for insertion sequence 6110. Now, I think you guys remember when I was talking about human genome annotation, that the human genome is just rife with repetitive sequences. Retrotransposons and ALUs and all sorts of signs and lines and all these great things. Well, we're not the only genome that has been exposed to repetitive sequences. That's also true in the insertion sequence 6110 stretch. This is found in many copies, and not just in Mycobacterium tuberculosis. It can also be found in stuff like nsMEG, I think. So I went back to the paper that documented the sequence of this very carefully back in 2008, and that's what the IS6110 looks like. So here's the trick. We found peptides that map to this, but those same peptides could be explained equally well by IS sequences that appear elsewhere in the genome. So this particular IS6110 was deleted in this species. We still detected the IS6110, but that same sequence is present elsewhere in the genome. So just because we identified peptides to this doesn't mean that this particular copy exists, because there are backups elsewhere in the genome. So this was no disproof that that region of difference exists. We had other parts of the genome that were present that could still account for our having seen those peptides. Remember that the peptides and proteins that we observe in proteomics do not tell us where their gene was located in the original genome. We just know the sequence. And that sequence may match to multiple possible sources in the genome. So Right. So in general, I would come to this conclusion. I think it's a pretty safe one still. One cannot prove absence through proteomics. You can only produce contrary evidence to absence. So in this case, we have some contrary evidence, but because we have additional places in the genome that could have produced it, we don't treat this as any sort of refutation that this region of difference was not actually missing. Okay. So this brings us to the takeaway message. We start with this concept of turnover. Protein turnover implies that messenger RNA and protein quantities may give different values, right? That the amount of transcript you see for a particular gene may be out of sync with what we observe in protein quantitation. You may have lots of copies of transcript, you may have very few copies of protein, or vice versa, because they are subject to different processes of synthesis and degradation. And those balances can be set so that very small numbers of messenger RNAs that are very long-lived lead to very large numbers of proteins because the translation of that messenger RNA happens again and again, for example. Lots of different regulatory ways that these things come into balance. Proteomics can confirm a subset of variant sequences derived through genomics. Now, I know that proteomics people are a little loath to say this, but I would note that when we do a genome sequencing of an organism, we almost always find a far larger number of variant sequences than we will ever detect by mass spectrometry. Mass spectrometry is a relatively insensitive method for screening. It's just the best that we've got. Shotgun proteomics can identify, tens of thousands of peptides from a sample, and yet it may be that the variant peptide sequences are being expressed, but we just missed getting a really good spectrum of it. So that's kind of an uncomfortable reality, but generally speaking, a genomic screen for variation is much more likely to detect a mutation than we are to see the translational byproduct of it. And finally, despite the deletion of a region of difference, peptides may yet appear via paralogy. We've talked a fair bit about orthology when we talked about bioinformatics. That is the same gene appearing in multiple species. Paralogs represent gene duplication events. In the case of this insertion sequence, the insertion sequence made lots of copies of itself throughout the genome. And so when we found peptides that could have mapped to the region of difference, we found that there are other locations that could have given rise to it. So that is a very, very quick appraisal of what proteogenomics has to offer in the space of mycobacteriology. You're joining a department that has published in this space before, and I suspect will publish in it again. Proteomics is valuable, even if you think of your research as principally genomic or genetic appraisal or biochemical appraisal. I think that proteomics probably has something to offer as well. So I hope that this has helped open your mind to some of the other things that might be possible. Great, thank you very much.